Hello, you're watching Armando Hasurungan Biology and Medicine videos. Please make sure to subscribe, join the forum and group. For the latest videos, visit Facebook Armando Hasurungan. Like, ask questions, answer questions, and please post some artwork, maybe. So continuing the metabolism map, we'll be looking at glycerol synthesis first from fructose. Now fructose is a monosaccharide. Fructose first converts to fructose 1-phosphate with an addition of a phosphate off the first carbon. The enzyme used in this process is fructokinase, and the phosphate group was obtained from ATP. Now fructose or fructose 1-phosphate can actually both convert to fructose 6-phosphate, which was part of glycolysis, but we will focus on a different pathway. So fructose 1-phosphate then splits in half to form two molecules, one of which is glyceraldehyde, and the other one is dihydroxyacetone phosphate, if you remember, which was part of glycolysis. Now this is done by the enzyme fructose 1-phosphate aldolase, not isomerase. This isomerase here is a mistake, sorry. And then glyceraldehyde then can be phosphorylated. It can be phosphorylated to form glyceraldehyde 3-phosphate by the enzyme triose kinase, and it obtains a phosphate from ATP. Alternatively, glyceraldehyde can be reduced to form glycerol. So it can be reduced to form glycerol by the enzyme alcohol dehydrogenase, and NADH is then oxidized to NAD. Now glycerols are pretty important. It is a sugar alcohol that is a backbone of many lipids, and it is also an important intermediate to carbohydrate in lipid metabolism. Now it can also be used in gluconeogenesis to produce more glucose when it converts to glyceraldehyde 3-phosphate. But we will continue and see how glycerol forms the backbone of membrane lipids now. So glycerol can then be phosphorylated to L-glycerol 3-phosphate by the enzyme glycerol kinase. Phosphate group comes from ATP, and it's added off the third carbon. Now L-glycerol 3-phosphate can also be oxidized to form dihydroxyacetone phosphate by the enzyme glycerol 3-phosphate dehydrogenase, and so can be used for gluconeogenesis. So if glycerol 3-phosphate was oxidized, this would mean that NAD is reduced to form NADH. But what we're focusing on now is that L-glycerol 3-phosphate can alternatively, it can become phosphatidic acid by adding two fatty acid groups on it. So L-glycerol 3-phosphate can become phosphatidic acid by adding two fatty acid groups. Now fatty acids as in acyl groups. So two acyl CoAs transfer two acyl groups to L-glycerol 3-phosphate to form phosphatidic acid, and this is done by the enzyme acyl transferase. So now on a side note, let's remember what acyl CoA actually is. Now I used to get confused with acyl CoA and acetyl CoA. They are obviously different. Now acetyl CoA makes acyl CoA. Now acetyl CoA was a product of pyruvate, remember, by the pyruvate dehydrogenase complex, and acetyl CoA is what enters the Krebs cycle. Acyl CoA looks like acetyl CoA but with a long hydrocarbon chain. That is why it is a fatty acid CoA. Now acyl CoA can be any number of hydrocarbons, and depending on the number of carbons it contains, it can be given a special name, such as palmityl CoA, which is a 16-carbon chain. Anyways, so we can even add more hydrocarbons to acyl CoA, and then by removing the CoA group, it can become a fatty acid essentially. And remember that, so it can become a fatty acid. And remember that acetyl CoAs is what makes acyl CoA or fatty acids. Okay, and the next thing we will talk about is phosphatidic acid. And what is phosphatidic acid? It is actually an important constituent of a cell membrane. It plays a role in the functioning of cells, such as cell signaling. It is composed of a glycerol backbone. And attached of the third carbon is a phosphate group here. And of the first and second carbons are ester bonded fatty acid chains. Now, this constitutes a phosphatidic acid, but if this constitutes another fatty acid, but if we add another group of the phosphate, it may change the charge of this lipid. And so with the head group, this is now known as a glycerol phospholipid. Yes, interesting. Now, going back to the map, phosphatidic acid can, as we mentioned, synthesize special glycerol phospholipids. So phosphatidic acid can synthesize special glycerol phospholipids by adding a head group of the phosphate on the third carbon. Now, depending on the head group, the overall glycerol phospholipid can have a negative or positive charge, which will affect cellular transportation in some way. Phosphatidic acid can alternatively dephosphorylate to diacylglycerols. So the phosphate from the third carbon can be removed. And the enzyme responsible for this is phosphatidic acid phosphatase, not dehydrogenase. This is a mistake. Now the phosphate group was transferred to ADP, forming ATP. And the phosphatidic acid, and this is a mistake. There's no NADH or thing. Now, diacylglycerol can then convert back to phosphatidic acid, also by the enzyme diacylglycerol kinase. So not phosphatase, but kinase. And it obtains the phosphate from ATP. Now, going back to diacylglycerol, it is a glycerol containing two fatty acid chains, conveniently binded to the glycerol molecule through ester linkage. If we remove a fatty acid from diacylglycerol through a hydration process, it will then form monoglycerols and a free fatty acid, a glycerol with only one fatty acid chain. Now, the free fatty acid are usually stored if they don't do anything. Now, this reaction is carried out by a special class of enzymes known as lipase. If the monoglycerol is hydrated again, it will break into a fatty acid, and if we follow this line, it will form, the other one is a glycerol, because monoglycerol is formed by one fatty acid and one glycerol. And the enzyme responsible for this, as we mentioned, is lipase through a dehydration reaction, a hydration reaction. If fatty acids needs to be packaged up, a fatty acid chain can be added through acyl-CoAs to diacylglycerols, which will form triacylglycerols, a glycerol with three fatty acids, acid chain. And the enzyme used is, if you remember, acyltransferase, which will transfer the fatty acid group from the CoA. Now, triacylglycerols can also be hydrated to form one fatty acid chain and also diacylglycerol. The enzyme used is hormone-sensitive lipase. Now, all these triacylglycerols and diacylglycerols come from adipose tissue, or lipoproteins. So that was it. So we learned about these fatty acids constituents, but how are fatty acids actually synthesized? These fatty acids, how are they synthesized? Now the next video will explore this topic. But first we will look at how... So fatty acids are made from acyl-CoA, remember? So acyl-CoA is in the mitochondria, but fatty acid synthesis occurs in the cytosol. So how does acyl-CoA get out of the mitochondria into the cytosol? This is the topic of the next video. And following that, we'll look at fatty acid synthesis from acyl-CoA in the cytosol. Thank you.